My name is Elizabeth Alexander and I'm the Chancellor of Melbourne University, and it is my great honour to welcome you to this celebration of women in medical research. I commence proceedings by acknowledging the traditional owners of the land we gather on, the Wurundjeri people of the Kulin Nations, and I pay my respects to the Indigenous elders past and present. And today's celebration takes place in the 150th year of medicine at Melbourne. Yet it is only the 125th anniversary of the admission of women in Melbourne as medical students. And today I would like to mention those early women who campaigned hard but successfully back in 1887 to gain admission to the medical profession through what was a very conservative school of medicine back then. Later generations of women have been inspired by their example, just as many young women are today inspired by the example of the many women medical scientists whose work we celebrate today. I was interested to read recently that in the past 3 years, women have outnumbered men in the Melbourne Medical School, and that we now have about 53% of graduates are women. This result comes about because of several things. One is the determination and strength shown by trailblazing women within the medical profession. Another is the mutual support and encouragement medical women have often shown towards one another. But the rise in young women studying medicine also comes about because of inspiration. Young women are inspired by the example of the success they see. And to just mention a few, what great examples of inspiring success we have here today, with Elizabeth Blackburn, Fiona Stanley, Ruth Bishop, Ingrid Schaefer, and I could go on and name many more. And it is certainly an honour to be part of it. It is particularly an honour for someone like me who comes from a finance background. As the first woman to be a partner in a major accounting firm in Australia, I can say that I have seen some of the hardships women face when they tread new paths within a conservative profession. Yet I must say the challenges I have faced seem very little compared with the challenges that have faced women in the medical profession, particularly in the early days of its history. So I salute the many women who have achieved spectacular things in medicine and medical research. You have achieved great outcomes for your fellow human beings. You've not only changed the practice of medicine through your outstanding medical research, you have also pushed back the frontiers of knowledge. You have also reached into the highest echelons of the medical world, and your success is underlined by today's event. It is an honour to be here with you, and it is a great pleasure to open proceedings by extending to each of you a very warm welcome to Melbourne University. So thank you.
So thank you very much, Chancellor. It's my great pleasure to be here to add the University's welcome to today's event. My name is Margaret Sheil. I'm the Provost, still called the new Provost of the University of Melbourne. And it's wonderful to be here and to be wedged between two great Elizabeths. I was telling my daughter Elizabeth this morning that was going to be part of my day. And to once again have the honour of sharing the podium with Elizabeth Blackburn. 2 years ago, we actually had a unique period in Women in Science in Australia where we had the President of the Academy of Science, the Chief Executive of CSIRO, the Chief Executive of the ARC, the Chief Scientist, the President of the Federation of Australian Science and Technology Society, and the Chief Executive of the ARC, which I was then, were all women. I don't think we'll ever replicate that again, because several of us have now moved on to other roles. But what was particularly special about that year was also that we had the opportunity for Elizabeth to visit, and we also had the honour of sharing a number of events with our first female laureate. And that collective group of women that I just referred to, a number of us spoke at an event at another university, which we won't talk about down the road. While there were some tremendous presentations, definitely Elizabeth Blackburn was the star of the show, and to see the school children and other scientists surrounding her after her brilliant presentation was a true honour, and I'm sure we're going to be similarly pleased today. Elizabeth obviously has strong connections to this university. She was a science student here at Melbourne, and like her mother before her, a resident of Janet Clark Hall during her study for a Bachelor of Science. She then went on to a Master of Science specialising in biochemistry, before which it's been described here as a classic academic career path. I think it would be better apt as a stellar academic career path, going from Melbourne to Cambridge, and then on to a postdoctoral period in molecular and cellular biology at Yale before joining the faculty of the University of California in Berkeley, and later moving on to the University of California, San Francisco, where she served as chair of the Department of Microbiology and Immunology from 1993 to 1999. Elizabeth is currently the Maurice Herzten Professor in Biology and Physiology in the Department of Biochemistry and Biophysics at UCSF, and she's a non-resident fellow of the Salk Institute for Biological Sciences. Throughout her career, Elizabeth's achievements have been recognised and honoured by her peers through many prestigious awards and fellowships. She's a Fellow of the American Academy of Arts and Sciences, the American Academy of Microbiology, and the American Association for the Advancement of Science. She was the President of the American Society for Cell Biology in 1998 and the American Cancer Association for Cancer Research in 2010. She's had numerous awards, the Albert Lasker Medical Research Award, the L'Oreal UNESCO Award for Women in Science. She was named one of Times Magazine's 100 People Who Shape Our World in 2007, became a Companion of the Order of Australia in 2010. Served on presidential committees, and if that wasn't enough, in 2009 she was awarded the Nobel Prize in Physiology of Medicine for her work on telomeres and the co-discovery of the regenerative enzyme telomerase. This research, in partnership with Carol Greider and Jack Szustak, with whom she shared the prize, had an enormous impact on understanding the effect of age and physiological stress on cells, and some of you will be much more familiar with that work than I am. She's been, as I said, an inspiration to many women, including myself, and we're truly fortunate to have the opportunity at the end of a rather long tour that she has indulged us here at the University of Melbourne to speak today on Don't Sell Yourself Short. Please welcome Elizabeth Blackburn. Thank you.
Thank you very much. It's truly a pleasure to have this opportunity to be here and to speak to this group. And this especially means a lot to me because I was a graduate of the University of Melbourne, where I received an absolutely excellent education, and which was one that enabled me to go on and do what's been a great joy in my life ever since, and that's to be part of the great tradition of biomedical research. And so I thought, rather than show you slides and things, I would talk to you about really what could I draw from my experience that's relevant to the topic that's being celebrated today, women in medical research. And so I think I'll begin with, a sort of a broader aspect of the kinds of considerations we have when we think about medical research. I want to use the example of my research to make various points, and then sort of end up with sort of trying to think about some rather more specific things that I've found have been helpful for me to think about and perhaps have helped others in medical research, particularly women. So I think it's a truism to say that biomedical research has become an integral strength in Australia, an integral strength of Australia. And clearly the track record is strong. But I think the question we all have to ask is, well, can Australia afford the status quo, and particularly the status quo as we see it at this moment as regards women in science, because it's obvious that for science to stay optimally strong and vibrant, what we need is to foster all of the best minds that we can bring in, not only to engage in research, but also to execute and think about policies arising out of research. And that, of course, means making sure we have women in medicine and research well represented, not only in the strong numbers we see represented in those who come into medical school, as was alluded to, but continuing to stay as engaged as possible. So, as I said, I'll try and draw from things that were in my experience. And my life after graduating from my PhD and postdoc really has been to be fully engaged in biomedical, as we thought of it then, biological, and now more and more related to biomedical research. And so the way it started for me was I just wanted to understand how biology worked, how nature worked, and through, again, my good education here, primarily at the University of Melbourne, I really got the idea that the molecules of life were going to be what was a way of understanding how life worked. So that was truly my curiosity about how nature was working. That was the driving force for me, and it certainly at that stage was not the broader questions of the implications for health or medicine of the kind of research we did, but really fundamental question. And the fundamental one in molecular biology was, well, what was at the ends of chromosomes? So let me just tell you, and I think it's familiar with everybody here, of course, that we all know the chromosome has a long linear DNA molecule, and it turns out the ends have to be protected. And cytological observations had said for decades that there was something protective at the ends of chromosomes, mostly defined by what was not there, that is to say, or what the ends did not do, and that was they didn't engage in the joining reactions that broken chromosomes engage in and elicit cellular responses in response to such breaks. But the ends were sort of inert in some way, and sometimes even a cytological knob, and it was sort of unclear what was there. And so from the training I had by going to Cambridge and working in the lab of Fred Sanger, where the DNA sequencing methodologies were being worked out. And can you believe it was actually interesting to sequence DNA in those days because nobody actually had done it. So I'd learned technologies which were then the laughingly simple to us now, but cutting-edge technologies of the time to how to get some inroads into DNA. I joined a lab in which the biological system that they were working on, produced a whole lot of very short linear DNAs, natural chromosomes, so I could actually analyze the ends of these using these molecular techniques. And so the first thing was, using cutting-edge technology and trying to ask a question that somebody else wouldn't have asked necessarily. That was the approach, and very much directed at a fundamental question in molecular biology. And then, as a result of observations on telomeres that Jack Szostak and I and others were making, we started to think maybe there'd be an enzyme, something that adds DNA to the ends of chromosomes. So again, breaking out of the textbook sort of stories and saying, well, if there was an enzyme, one would have to roll up one's sleeves and actually try and see biochemically if there was one, drawing from my good training in the biochemistry degree at the University of Melbourne, thinking about enzymes and how you might, approach this question. So basically, Carol Greider and I in my lab at Berkeley then discovered that there was an enzyme called telomerase which replenishes telomeres, adds DNA to the ends. And so without the telomeric DNA and its special way of replicating which involves telomerase, it became clear that chromosome ends dwindle away as their telomeric DNA erodes, and eventually that causes cells to stop dividing. And to fast forward, and now thinking about the work of many, many groups around the world and many, many different approaches, it's fairly clear that for humans to have a healthy, normal, long life, one has to have this dwindling down of the ends appropriately counteracted. And if that doesn't happen, that has consequences in terms of disease processes as they develop, particularly in adults as they get older. And so now, of course, the challenge is to develop emerging molecular and cellular information of the kind that I've just been talking about, involving telomeres and telomerase and now a lens on the world into rational ways of improving human health. So that would be, one of the bigger questions. So So like all questions in biology, and this is just one example of one kind of thing, you know, you look at large, complicated problems like those in science and technology, and it's fairly clear, I think, studies have shown that problems are solved better by input from a lot of different viewpoints. Now, it may seem that problems can be solved, you know, more quickly and more effectively by one group who are all thinking alike, they sort of work along the same lines and so on. But actually, you know, for the complex issues of the world, and science and technology, as I said, exemplify this, the quality of the solutions, it's pretty clear, can suffer from what's called groupthink. And in that way, critical inputs lost, aspects of the issue can be overlooked. And so rather, it seems that long-term solutions to complex problems usually gain by diverse input from those with diverse views, diverse, you know, life experiences, diverse ways of seeing questions, diverse ways of approaching and dealing with issues. So in other words, what I'm getting at is I think we can't leave out women as players in science and technology, not least for that kind of consideration. And again, I want to draw from my personal experience in science and reflect a little bit about, you know, I'm not sure if a scientist who is a man would have actually done particularly some of the later work that we've been doing that I'll tell you about, but even some of the earlier work that we did, if they would have necessarily done or initiated the work that we did. I sometimes wonder about that question. And so again, let me give you a little of the setting and why I wonder this kind of thing. So for example, after we discovered the existence of the enzyme telomerase, many researchers found that in humans, telomerase was very noticeably active in cancer cells. And so there was this idea that cancer cells, which of course are very different from normal cells in a whole lot of ways, somehow, you know, maybe they had telomerase, but in adults, there was no telomerase in normal cells. So there was this sort of very digital view, yes, no, that cancers would have telomerase and normal cells or many normal cells in adults, in humans, wouldn't have telomerase. And so that actually, you know, was one viewpoint. But in fact, there's a subtlety there, and it sort of meant approaching this question in a different way. Because in my lab, we said, well, maybe that's not true. And we started looking and started finding that actually in normal cells, the kinds in which, if you read reviews that said telomerase is turned off in normal adult human cells, right? And that was sort of this groupthink. But if you looked, it was subtle. Telomerase is actually present in low and quantifiable, and it turned out meaningfully quantifiable amounts in normal cells. So again, I think sort of being prepared to go outside what becomes groupthink and the, you know, what's the leading paragraph in reviews on subjects is important. And I think that women, and again, you know, grossly generalize in some way, but that's my sense, is perhaps there's a, you know, a sense that people do sometimes think outside the box if you're not necessarily, you know, in the majority group of people, which in the case of the more senior science sort of leaders often tended, at least where I was working in the US, to be men rather than women. Okay, so, and then another kind of question has been, so what has emerged actually is, despite the fact there is this nuanced amount of telomerase, is that, in fact, there is a general observation, if you sort of look at 40,000 feet up and look at humans in general, there's a general observation that telomeres do shorten with age in an observational way where you just look cross-sectionally at one time point per person. So that had been part of the data that said, oh, well, there's no telomerase in normal cells, and it's going to run down. And again, as I said, that's much more nuanced. But again, we approached things in ways that I think were probably affected by the fact it was a woman scientist actually approached me back in about 2000. Her name was Alyssa Eppel, and she was in a completely different field. She was in the field looking at chronic, severe psychological stress, long drawn out kind of the kind that's often associated with very sort of severe situations. And I'm going to give you some examples, okay? And she said, well, what happens to telomeres? You know, very different, and telomere maintenance and telomerase, you know, just a very different way of approaching this question. And I didn't know, and I said, I don't think anyone knows. And so we started a series of collaborations, which have then grown out into a whole lot of others. But particularly, what we found, just by being prepared to look and ask the question, was that in caregivers of a chronically ill family member, initially it was with, these were women caregivers, initially where it was her chronically ill child, her biological child, or in later studies we're still doing where it was a dementia patient who was often her husband or spouse, often with Alzheimer's. Oh, this goes on for a long time. And basically what we quantified was the severity of this, the chronicity of this, with shortness in telomeres, and actually sort of dampened down telomerase, which is what replenishes ends, so therefore dampens down the abilities of the protective telomeres to be maintained. We found both of those quantifiably correlate with the severity and duration of the stressors, measured in a variety of different ways. Now again, this was something where I have to say, I think about the fact that I'm a mother myself, and our son was, a kid then when these studies were started, and I could have sort of had I could imagine how it might have been for a mother to be the mother of a chronically ill child for whom she takes primary caregiving responsibility. So I particularly sort of related to that, although I've been spared such a severe situation myself. I could imagine how it might be. So I just wonder, if you choose things somehow based on your life experiences. A woman clinical researcher colleague initiated a study with us, and she's been studying women who had suffered abuse formerly by a spouse or a domestic partner, right? And so again, we quantified telomeres in them, and in a study we just published not too long ago, we found that indeed, yes, the telomeres are actually shortened, and that is related quantifiably with the fact that they're in this situation, and the shortness even persisted 4 years after the abusive situation had stopped. So again, these are starting to be things reaching out into lives of people, and quite interesting thought is who chooses to study these kinds of questions. Again, I have not personally had that situation, but again, one's imagination can sort of encompass what such things might mean. Again, thinking about topics, one of the things that we've been doing, and again, it's interesting that a lot of these studies are done with female collaborators, and of course not only female collaborators, but they've often been initiated by them, looking at effects reaching back in childhood. So we're now finding that severe stress on a mother is actually reflected quantitatively in shortness of telomeres in her children, and when the mother has had this stress while pregnant. So we're looking at developmentally imposed effects during gestation. So again, interesting things we're finding. Shortness of telomeres quantifiably related to adverse social and family situations in the children now, and shortness of telomeres in adults who in the past have had severe family trauma type of situations, adverse childhood events of severe kinds, which happened decades before, but quantifiably the numbers of categories of such childhood trauma or adverse events, as they're called, is quantifiably related to telomere shortness in the adults when they're well into adulthood. And this has been seen, again, in various studies. Now, again, I want to emphasize, of course, men and women are engaged in these studies, but I think it's interesting that I look quantitatively, if you will, at who has come to us and initiated these studies, and they very often have been women researchers. And so my only point is not to say, oh, women will only do things and choose topics differently from how men do, but I just want to raise the question, well, do women initiate investigations into questions that might be different from those their male colleagues choose to initiate? In fact, do different experiences and viewpoints in life influence what kind of science gets done? And then in turn, that opens up fields which then broaden in much different ways. And so I think the relevant point here particularly is that what has been seen in a great many studies in different cohorts around the world is that telomere shortness is quantifiably linked to risks for the major diseases that are the major killers of the elderly in developed societies, such as Australia and so on. And so these, of course, are cancer and diabetes and cardiovascular diseases. But those major killers of the elderly all quantifiably relate to telomere shortness. And it's clear from rare genetic situations that reduce telomerase, and while they're much rarer, it's clear that they also have causal relationships in making genetic diseases that make less telomerase, shorter telomeres, and they have causal relationships with a set of diseases that overlap with and include the major diseases of aging, such as those I mentioned. So this sort of thing where we're looking at now what might have seemed a different kind of science, if you will, science that involved social issues and so on, in which both men and women are engaged. I think the questions that I've been asking may well have been affected by, as I said, the life experiences and so forth of men and women. So, just a thought here. And I think it's something to think about, because, again, my point really is complex issues. It's good to bring all sorts of different approaches in, and this is just my very own sort of personal experience here, but it was interesting when I think about it. So, we do need to have the best minds in research. Include everybody who can bring different views. Clearly that must include women. And so that then leads us to the thought, well, okay, what's the playing field like for women in research? Is it ideal yet? Because we do want the science and technology progress to be optimal. We want the workplace to be one that welcomes women, and that means not apologizing for, wanting that to be the case, because, as I say, I think it's very clear that science needs this diversity of brain power and experiences to come into it with different approaches. Well, let's think about the playing field, and I'm not going to give statistics, and I think you're all well aware of statistics and so on, but really just talk about some considerations that won't be unfamiliar to probably all of you here. Well, of course, we know the playing field is better than it was before, right? In fact, there's a lot of progress that has been made, but I'd say we're not quite there yet. What's the progress? Well, the progress is that there's emerged a lot of understanding of the issues that are important for ensuring that playing fields are level for women not only to enter science and medicine, but also to progress in it and do well in it. There's been a lot of progress in how to address these questions. At a lot of institutions and universities, it's been impressive to see ways that have been thought up and how to address these issues. But, I think that we're probably 10, 15 years away from getting to perhaps where we want to be. And I'll just quote somebody who's thought a great deal about these issues, a biomedical researcher, Nancy Hopkins of Massachusetts Institute of Technology, MIT. And she said, the pipeline of women in research has grown, as we heard, more medical students, women than men, I think, in Melbourne University now. The pipeline of women in research has grown, and people thought that that's all it would take. They thought time alone was going to solve this problem. Clearly, time alone did not solve it, right? Even after women were let in, invisible barriers keep them from rising from the top. So a generation of women had to get through the system and identify what the problems were, and then partner with institutions to find institutional solutions to those problems. For example, going way back, the first problem they encountered was sexual harassment, and it took 10 years, she's talking about the United States in this case, to figure it out and make a law prohibiting it in workplaces. I go on to quote her, generally when you identify a problem, it takes about 30 years to first identify, devise solutions, test the solutions, and then make sure everyone learns about it. Then there was a lack of mentors. And then there was one and this is Nancy Hopkins speaking still, one we worked on at MIT, the undervaluation of women for equal work. This is now called unconscious bias. And Nancy says, we're about 15 years in on that one. I think one remaining problem is we didn't know early on really how to address family issues. How do you get people to very high-level positions and make it possible for both men and women to have families? So I think that really has emerged as sort of the elephant in the parlor as we look at women coming from tremendously good training from institutions like the University of Melbourne and what happens next. And so there are various sorts of ways this can be approached. So I'd like to just go through my thoughts on them, first of all. So first of all, leadership from the top, and that can be so important. And I'll give you one example. It's very homely seeming, but I was very impressed when Shirley Tillman, who is the president of Princeton University, herself, by the way, a biomedical researcher, I was very impressed when she initiated a simple seeming and very effective childcare option. She went to the source of what's the biggest problem. It's when the childcare situation won't work, when a child gets a fever and they can't go to their childcare situation and the person has to give a lecture or meet a grant deadline. So what they did was the university partnered with a bonded, appropriately good childcare organization, that supplied 24-hour childcare and heavily subsidized it. So there was very little inertial barrier for somebody to be able to tap into this. They pay a small amount, but the university heavily subsidized it. It was a bonded, respectable, licensed childcare organization, not some fly-by-night thing. And so you could leave your child for whatever it took, the 24 hours or something like that. While this emergency situation, a deviation from your normal childcare situation, which you had so laboriously set up, while that need was there. Very simple example, but just as a very simple example. Also, things like, and I'm sure this is true here, that now if you have childcare leave when your child is born or you adopt a child, that in fact you don't have to ask for it. It's actually the default. You have to ask not to have that. So those are very simple institutional things which sort of take away inertial barriers to things that can seem like overwhelmingly difficult situations when women think about how am I going to manage all the demands of my career, and we all know what they are and how important it is. So this, this need that we feel, and I think appropriately, to have some balance in our life is something that we feel sort of often overwhelmed in early careers about how on earth are we going to do that. I heard of a terrific institutional solution from, of all things, an astrophysicist here in Australia, up in Sydney. He said a great thing. He said, every position we advertise as either part-time or full-time. Not that you have to go and apologize and say, I'd like to do it part-time. No, he said, we just do that routinely, men, women. And he said they get terrific people. And when you think about it as an institution, you can actually do some very good things. When there's two people, you sometimes get more than the sum of the two parts. You can imagine if they're related subjects, and you can imagine. So I think this is terrific. And he said, this is what we do routinely. It's simple. I mean, it seems like this is very, very obvious sort of stuff that as institutions we should be all thinking about. And then this question of balance, and I think the advice that I like to give to people as they're beginning early in careers and thinking about, oh my gosh, if I'm going to succeed, it's going to be this enormously heavy duty work, and how can I possibly have a balanced life? And I think, so first of all, arranging flexibility, and as I said, institutions can do a lot for that. But also something to think about is that it doesn't go on forever, the sort of intense needs for input with the family. There are periods, months or sometimes years, when certainly there's intense needs of the family or intense needs of research, but they don't have to happen forever. Both of them all at the same time. And sometimes one can sort of, do sort of waves in and out of one versus the other. Meryl Streep said every successful woman's life is a series of ebbs and flows, because she had a family and was a very successful actress. Now, that might be harder in our careers, but again, I think it's not impossible with input from institutions as well. And I know that I think my life was sort of very unbalanced for many years before my family started. In my case, it happened to be in my late 30s, but that doesn't have to be a pattern for everybody. And that was very out of balance. I was completely in science. And then I sort of joined the human race and somewhat and had the family. But the research went really well. And I had, a son was born, and family life is very important. But again, you sort of had periods of intensity both ways. And now he's grown up, so, now I can go to movies again, right? But, it was okay not to go to movies for about 18 years, except really lousy children's movies. But the point is, balance can perhaps happen over years. That's something to think about, because it often seems overwhelming for a young person to think, how can I possibly have a successful career in biomedical research or in the medical world? And also, be a human being and be able to be part of life as a human being in all its different dimensions. And the situation of Janet Rowley is a very illustrative one here. Janet Rowley, some of you may or may not know, was one of the early discoverers of the importance of translocations of chromosomes in driving certain cancers, particularly leukemias in the earlier days. And she won a National Medal of Science in the United States for this research, and National Academies of Science and Memory, very, very esteemed, very successful scientist. What she always tries to remind us all about is she did that work for which she is so recognized while she was a part-time researcher, mother of 4, bringing up 4, children, and she was doing it part-time. She even said there might have been a certain advantage to this part-timeness because you could sort of a little bit separate away from the work and perhaps your brain would process things a bit, I don't know. But she says she feels that being part-time might have been important for her to have been able to do the work in the way that she did it. So here's a situation that might have been seen as a sort of, quote, disadvantage, in the sense that she was only, quote, working part-time, but this work was done in that situation. So universities, institutions, I think, should be encouraged to have flexibility and creativity of the kinds that I was talking about. So, this does mean making workplaces user-friendly in ways that we've talked about, not only in terms of attitudes, but really in terms of, as I said, structural things that really, when you think about them, are not all that difficult to envisage and might even turn out to be advantageous, but just different from the ways they've been done before. Now, another thing that I really think about as I experience in my day-to-day life as an advisor, as the leader of a research team, as a collaborator with research teams and other researchers, I think I find something which is very corrosive, and I see in the young researchers a real eroded self-confidence, which is a very interesting thing, and we can all think about what might be the causes of it. But I see that particularly in the young women researchers, superb trainees who I would say have every ability to go on and do research, and yet I see examples. I'll just tell you one example, because it can often come from places that are unexpected. So this was a postdoctoral fellow in my lab, and she said, was I good enough to go on and do research? And I said, and have a position, as in the US it would be the case, an assistant professorship. And she said, was I good enough? And I said, yes, of course. And then she started to tell me that her brother-in-law, who also was in biomedical research, and he himself was hunting for a position and having difficulty getting it. And he said to her, oh, you don't have all these Science Nature papers, you're not going to get a job. So great, your own family, So, no, seriously. And I think he might have meant it. Okay, he might have thought, look, if I can't do it, well, she can't do it because she didn't have, as many publications as I do at this stage. So I think that can be very insidious, and I don't think it was necessarily malevolent, but that can be a very insidious thing. And so I said I'm sure she could. And then she did something very practical. And I urge all universities and institutions to do this, and I'm sure you do this here already. She took advantage of a leadership course for postdoctoral fellows. It was actually organized by our postdoctoral fellows society on campus. And it was a one-year leadership course, and it was all about what is it like being a leader, and particularly tied to a leader of a research group, which is the relevant leadership for the kind of thing she was looking for. And what she got was she got sort of a toolbox, if you will, to deal with the immediate challenges. And she had to involve me at one point because she had to, you know, how did she deal with me versus others? And so I learned a little bit about what she was doing. What was fascinating was the transition I saw in her. And, you know, it was very tangible and very, very interesting because she realized, oh, she could do this. And so, indeed, she's thriving as a, you know, in her position at a good university as an assistant professor. But, you know, so a very practical thing, a one-week course on leadership at the university through the postdoctoral fellowship organization. I'm sure there's similar ones here. That really just, it turned her around, I think, in a lot of ways. So obviously, the other sorts of things I think about are, you know, what's the useful kind of advice, again, I could give from my kinds of experiences. And certainly, we all know that as scientists, our brains have become extremely good as researchers, as medical trainees and clinicians. Our brains have become very good at solving problems, thinking about problems and solving them. And sort of the idea of using that to solve one's very personal-seeming problems of how does one deal with life and family and all those things, you know, often seems a little foreign to people. But when you say you really got this great this great problem-solving part of your mind there, and it's, you know, it's okay to use that for something, you know, related to sort of love and family, which somehow might have seemed you couldn't use that part of your brain for that. People sometimes say, oh, yes, yeah, actually, you know, now I could think of solving problems in this kind of more analytical way, which sometimes people thought did not really belong in that more emotional part of their lives. And of course, you know, engaging with other researchers and scientists and clinicians in one's peer group and talking through all of these issues. Now, personally, I was very bad at that, and I think I sort of needlessly suffered because I felt too proud to admit that these might be problems. And I would say, you know, don't do that. You know, especially, I think, there are incidents that happen even as one grows through the seniority ranks. And this is something important to think about, that it's not just in the young, early career people, but all through there are situations one finds out about where sort of another, you know, a senior member of one's science community or faculty member or institution or something, there are cases where this individual will try and intimidate a threatening-seeming woman peer. And actually one would be surprised that this happens surprisingly often. And one only learns this when one talks to other women peers, and then one starts finding out, and sort of, you know, that depersonalizes some of it, and one realizes that there are sort of workplace and attitudinal things which, as I was saying, you know, we still have a way to go with. So certainly engaging one's women colleagues, but also very important to engage one's male peers as well, and using, you know, science and approaches, sometimes depersonalized ones, that can help to persuade. So that's, I think, important kinds of lessons I've been learning, not in the very early career stage only, but also continuing through, because that's where one finds fewer and fewer women. And so indeed those kinds of things, which used to be issues, you know, in the very young, and now that's not an issue at all, with half the, you know, class being women in medical school and biomedical sciences up to the PhD and advanced degree levels. So these things are still somewhat going on. So I think the last thing I'll say is that one of the things that I have learned from all of this is, you know, I almost sort of think of it as the M word, because mentoring, it sounds like such a cliche, but actually in all of its different meanings is a very, very important thing. I've been the recipient of it in various stages of my career, and I think there's, you know, nothing that can overstate how important this can be. And that takes, of course, different forms depending on where you are and what stage you're at. You know, as I say, mentoring in terms of your peer group, this can be very helpful to a certain degree. But I think mentoring from other people at different stages is very crucial. And and I think that all of us really should reach out and try to mentor whenever we can. And as we all know, and I think we've probably all had this advice, there's sort of mentors for different aspects. Some mentors are very good for some aspects, some mentors are very good for other aspects. There isn't one mentor per se, but people who really can be there and draw out of you what it is you need to draw out of yourself in the end. So that's really what I wanted to talk about. And I think if I had to have a message for everybody here, I would think about this way of saying it. First of all, I'd say, well, take heart, right? And let yourself rage a little bit if you're beaten down. It'll always happen at certain stages, right? And then you have to pick yourself up, right? And think about it analytically, but think about it honestly. And finally, bring some laughter to bear on your next steps. So let me thank you for letting me talk to you at this wonderful afternoon and wonderful occasion, and thank you.
Thank you very much, Elizabeth, for what I think was, truly inspiring. I myself learnt some more interesting things that I didn't know, and we'll take some of your advice on in some of our work. We're going to have a facilitated session later, so there'll be opportunity to ask you questions then. So I'll conclude mine and hand over this session and hand over to Jane for the next one. Thank you.
Thank you, Margaret. And what a wonderful opening to our afternoon. And we're in for a great afternoon, and it's great to see everyone here. I'm Professor Jane Gunn. I'm head of the Department of General Practice, and I've been part of the Melbourne Medical School 150 Years organising committee. And we're delighted to have this event on this afternoon, and especially with Elizabeth being here. We've heard some really interesting things from Elizabeth, and now we have three closer to home currently women that are going to share their stories with us as well. And this is three short presentations. They're each for about 15 minutes, and then we're going to invite everyone up the front and you can ask questions. So to begin with, I'd like to introduce Professor Ruth Bishop, who will be our first presenter for this afternoon. And Ruth, I'm sure, is well known to many of you. She's an eminent Melbourne scientist who's devoted her life to improving child health. She is a graduate of the University of Melbourne with Bachelor of Science, Master of Science, PhD and Doctor of Science, all awarded from the University of Melbourne. She's received many prizes. I won't go through all of them, but in particular, she has received the Children's Vaccine Initiative Award, which was issued by the World Health Organisation in 1998, and she is an Order of Australia Medal in 1996. Ruth's best known for leading the team, which in collaboration with Dr. Ian Holmes at the University of Melbourne's Department of Microbiology in 1973, discovered rotavirus. And certainly as a medical student here, I remember learning all about rotavirus and how we were the home of rotavirus, and rotavirus is a special part of every medical student's education here. And Ruth, for those of you who didn't know it until this moment, is one of the reasons that we have that wonderful knowledge passed on through every graduating cohort. Now, Ruth has vigorously pursued the goal of preventing a very serious infection, and for any of you who've been to the Third World in particular, you know just how devastating rotavirus can be, but just as devastating on the paediatric wards at the Children's Hospital. You will find children very, very ill. Now, this work has made Ruth an international icon in the field of diarrheal diseases in children. She served on and chaired several World Health Organisation committees and has given wise counsel to many WHO programs. And I'm delighted to welcome Ruth this afternoon.
Thank you very much, Jane. I really enjoyed Elizabeth's talk. I think she laid down some extraordinarily interesting principles for us all to discuss later in the afternoon. My account is going to be a much more personal and chronological account, I think, and you can take from it what you will as to just how to keep going. I was given some headings to talk about, and this was the first, the appeal of science. It was asking me, did I always want to be a scientist? And when I really thought about this, and what I felt at 16 when I had to decide, was that initially I had no positive drive towards a career in science. I had a negative drive towards careers I didn't want, and a Bachelor of Arts and teaching was one of them. But as a result, I embarked on a science degree at the University of Melbourne. Through those 3 years, I also ruled out things I didn't want to be. I didn't want to do mathematics, I didn't want to do physics, I didn't want to do chemistry. And I was really left with the medically oriented sciences, microbiology, and extraordinarily the year that I was to do my final science year, pathology decided they would enlist science students. So I became one of the 2 pioneers of pathology done in a science degree at Melbourne University. I enjoyed pathology enormously. I loved the detective side of it. I loved being presented with slides and sections and deciding what might be the diagnosis. But I was clear-sighted enough to realise that there was really, at that stage, a very difficult course ahead of scientists who wanted to do pathology, so I focused on microbiology. The creative spark was a title that was given to me, and I really have had difficulty working out what this is. Did I ever have one? And if so, when and how? And I decided that probably the closest I got to it was when, as a second year science student anyway, I was in a few lectures where I found the lecturer incomprehensible. And I thought to myself, I've got to pass this subject. And so I repaired to the libraries and I taught myself a lot from textbooks. And I think in retrospect, this was a really very important thing for me to do, because it showed me in a lot of cases what was missing from textbooks, and that there was lots of scope for questions and lots of scope for trying to work out answers. And so I guess that's where a creative spark, as I said, if I ever have one, came in. The hard slog. As a final year student, I accepted an invitation to become a research assistant to one of the professors in the microbiology department. And he failed to get a grant, which was a very, taught me one of the first lessons associated with a research career, is that you can never be certain of finance. And if you miss out on it, you've just got to look to an alternative. I accepted a position in a diagnostic laboratory, but I quickly realised it was not for me. There was a lot of pipetting, sitting down having a cup of tea until the alarm went and putting in the next lot of ingredients into the tests. And I thought, this is not why I've spent 3 years doing a science degree. Fortunately, Dr. Sheil Anderson at the Royal Children's, who was interested in the microbial flora of children with malabsorptive states, had lost her research assistant. Her husband had been sent to Switzerland, and a fully funded project that had been embarked on in the department was without anyone to do it. And she approached me, I accepted, and in fact that set the path for my entire career. It was accidental. I had replaced her research assistant, and as a result of working first with Sheil Anderson, learning how to grow and identify microorganisms and associate the abundance of certain species with malabsorption, I eventually wrote an MSc. I was then recruited to go down to the Department of Surgery in the Alfred Hospital, where they were interested in intestinal obstruction and the role of intestinal flora in vitamin B12 deficiency. And out of that work, I wrote a PhD. I finished writing my PhD 4 days before my first child was born. And he was photographed, aged 9 months later, held in my arms after a graduation ceremony. The photo appeared in the Sun newspaper under the title, "Mum has a PhD." He tried to replicate this many years later when he got a PhD and I attended his ceremony, but it didn't kind of work. I suppose I now embarked on what you'd describe as the hard slog. I was very interested in hearing Elizabeth say that part-time work was a good solution, because throughout the next sort of 6 or 8 years, I think I had part-time funding anyway. I continued working at the Children's Hospital, again, intestinal flora and malabsorption. However, if you ask my husband to honestly say, did she work part-time, I think he'd say no, because I might have been paid part-time, but in fact, the other part of my work, once the children were in bed, started at about 10 o'clock at night and went through often until 1 or 2 in the morning. There's quite a lot of time in your day, if you really look for it, for reading and writing. I did, after a while, was again approached by Charlotte Anderson at the Children's to go back and do a project on, again, intestinal flora in children with malabsorption. But very soon after that, she got a job as Professor of Paediatrics in Birmingham, and she was replaced by Dr. Rudge Townley. This was, although I didn't realise it, a very critical moment in my career, because Rudge had a technique which was novel worldwide. He could intubate children and he'd modified the tube so that it didn't sit in the stomach and take 2 days to go through the pylorus. He could manoeuvre it through the pylorus within a few hours and provide specimens. This actually opened up a whole lot of different scenarios for obtaining specimens from stomach and small intestine and for me culturing them. He also changed the gastro unit, which was almost entirely research-oriented, to the gastro department and took on the the ward which admitted children with acute gastroenteritis. This in 1971, when the project began, was a very severe life-threatening disease. You can see a child here, photos taken by Graham Barnes on admission to hospital on the left, 10% dehydrated and close to death, and 48 hours later rehydrated and the fat, happy, smiling child she really was. These children were admitted in large numbers, particularly in the wintertime, to the Royal Children's Hospital ward, gastro ward, and a microbiological cause could only be found in about 30% at the most. So there was a missing 70 to 80%. Clearly it was infectious. It was most likely a virus, but all attempts to grow viral viruses from faeces had been unsuccessful. This is Raj with a child who's he biopsying. At that time, Graham Barnes joined the department and embarked on an MD, and his task was to biopsy children and look for the intestinal lesion in children, and my task was to try and culture bacteria from the small intestine, which had not really been attempted in children with acute gastroenteritis. The result of this research was that I was unable to culture anything that could act as an etiological agent, but Graham had identified the lesion in the duodenum. Left is normal duodenal villi, right is an acutely infected and portion of a duodenum has lost its villus structure, full of inflammatory cells, and clearly in a very damaged state. Graham went back to New Zealand, and I was left with a puzzle, which was what was causing this massive small intestinal damage, and decided that there was evidence that perhaps there was a viral pathogen. Perhaps if we got some more tissue and looked using an electron microscope, we could identify viral particles. Some had recently been described in the United States as small virus, and in fact I think that was what I thought we would find. I wrote out, again, the dining room table in the middle of the night, the object of this research, to look for viral particles in cells of duodenal mucosa from infants with acute gastroenteritis. It was an optimistic aim, but in fact it bore fruit almost straight away. I recruited Ian Holmes, who was an expert electron microscopist and virologist in the Department of Microbiology here at the university, and a technician of his, Brian Ruck. We obtained duodenal biopsy and sent it to Ian, who had a look, and immediately in the first section of the first biopsy he looked at was a new virus. It's the team, Brian Ruck, myself, Ian and Jeff Davidson, celebrating some 10 years later. And this is what he found. I still think it's one of the most beautiful pictures I've ever seen. You can see the virus budding from the cytoplasm of the cell into the interstosis of the cell, and eventually going out and destroying the cell in the process. We fortunately located veterinary people in Melbourne who were actually finding an almost identical virus in calves at the time. And they taught us how to look in faeces, so we were able to move very rapidly from duodenal biopsies to fecal extracts, and this became the basis for epidemiological studies for many years afterwards. Again, I sort of describe rotavirus, which is what it was called, as something almost I fell in love with. It was the last thing I thought of at night, and it was the first thing I thought of in the morning. Certainly it was all absorbing, totally, and it linked me in with a worldwide group of people who probably gave me a bit more credit than I deserved, said, how did you do it? And I didn't really quite know myself, but I certainly, in talking to a vet, and said how embarrassed I sometimes felt as being regarded as a virologist, because all my experience was in bacteriology. He said, now you've got to earn your stripes. That actually is what led me to eventually become an independent scientist. We did, it was a hard slog. We had to learn numerous techniques. It was quite exciting looking back, because we did this at a time when technical abilities in the laboratory were changing rapidly. There were ELISA assays that were being set up, immunofluorescent assays. There was electron microscopy. There were new monoclonal antibodies. We enlisted assistance with all of these new techniques, and in addition had links with people overseas and people coming to our lab and people whom we sent to overseas labs. I was recruited to WHO, who had a control of diarrheal diseases research project at the time. They were instituting and educating countries to use oral rehydration to make the salts and then use them. And we linked into those and set up etiologic projects throughout much of Asia and Africa. And the answers came back very similarly everywhere we looked. Rotavirus was the cause of 50 to 80% of acute severe gastroenteritis in young children under 3 throughout the world. I was, after one publication, interviewed for the local paper, and of course journalists always want to know when you're going to introduce a cure for the population, when would there be a vaccine? And in 1973 I said 5 years. 40 years later, we now have at least 2 safe, effective vaccines that are in use throughout most of the developed world and are slowly being introduced to the developing world. I've had a remarkable career. Looking back, I think much of it was luck and much of it was hard work. I'd like to just leave you on that note. Thank you.
Thank you very much, Ruth. So we'll move right along to our next speaker, which is Professor Ingrid Scheffer. And Ingrid is Chair of Paediatric Neurology Research at the Department of Medicine at the Austin Hospital and at the Florey Neurosciences Institute. She also has had a wonderful year this year and was won the Women in Science Award, the L'Oreal UNESCO Women in Science Award, and had a wonderful time in Paris earlier in the year, which many of us heard about. So Ingrid is a paediatric neurologist here. She's worked together with Sam Berkovich and has led the field of epilepsy genetics research over the last 15 years in collaboration with the Women's and Children's Hospital in Adelaide. Now, as many of you will know, this collaboration has resulted in the identification of the first epilepsy gene and subsequently 9 of the 16 genes currently known. So Ingrid has described 5 new epilepsy syndromes and continues to work on genotype-phenotype correlation, the outcome and new treatments of epilepsies. She's recently been awarded the 2007 American Epilepsy Society Clinical Research Recognition Award. And her research interests are really around the genetics of epilepsy and febrile seizures, epilepsy syndrome classification, novel anti-epileptic therapies, and added to that, autism spectrum disorders. So welcome to Ingrid.
Thank you very much, Jane. I thought I'd change tack after those two wonderful talks and focus on clinical research and is it real science? And I know that what is clinical research, it may not be immediately apparent to all, so I thought I'd start with an example from my own research story. And just as a few introductory slides, I've worked in epilepsy, as you've heard, and it's a common disorder affecting 2% of the population. And if you look at children, a further 3% have febrile seizures. Seizures are due to abnormal electrical activity in the brain, which is what we record with the EEG or the electroencephalogram. There are many, many types of seizures, and most of epilepsy begins in childhood and adolescence, which is why it's so relevant to a child neurologist. And there are also now a whole host of epilepsy syndromes, which is what we try and diagnose when we see a child with epilepsy. So there are many types of epilepsy, but to break them down into two broad categories, we have the generalised epilepsies, where you have an abnormal network involving both sides of the brain, and the other group are the focal epilepsies, where the discharge tends to remain in one part of the brain. Now, prior to the work I'm going to tell you about, generalised epilepsies were considered genetic, and focal epilepsies were considered largely acquired disorders due to, say, a tumour or a scar on the brain. And in fact, here you see the most common of the scars associated with focal epilepsy in both children and adults, where the focal epilepsy can't be controlled. And this is due to this abnormality here of the temporal lobe with scarring of the mesial part, the middle part of a temporal lobe. And so people said, well, focal epilepsy was an acquired disorder. So enter this little 4-year-old girl, and she's having a seizure from sleep. In fact, she does this 8 times every night. And they're very dramatic attacks and very distressing. She has retained awareness through them. You can see she's quite frightened. So the whole thing lasts 23 seconds, but as I said, she does this 8 times a night through the night. And on her own, she's interesting, but what made her more interesting was her family history. And we acquired this family in 2 separate halves, and for the PhD students here, a year of my life later, we had a pedigree like this. And so the pedigree and studying this family and a few other families led us to describe this disorder of autosomal dominant nocturnal frontal lobe epilepsy. And here, usually beginning in childhood at around 8 years of age, the child has clusters of seizures that occur through the night, often waking from sleep with an aura or warning, and then they have these frenetic hyperkinetic motor seizures where they may even propel themselves out of bed, or a tonic seizure. The EEG is usually fairly unhelpful for this disorder, and many of the individuals in these families thought that this was either normal behaviour, a parasomnia, a sleep disorder, or even in some cases had been diagnosed as having hysteria because of the retained awareness. And as you could see, it followed an autosomal dominant inheritance pattern. So from this family, we worked with our molecular genetics colleagues who mapped the family to chromosome 20q and went on to find the first gene for epilepsy in 1995. And the gene, surprisingly, was in the neuronal nicotinic acetylcholine receptor in the alpha-4 subunit, and the actual mutation was right here at the ion channel gate. And this paved the way for us to understand that many genes for epilepsy are actually disorders of ion channels. So that's an example of how clinical research can lead the way in our understanding, working together with our basic science colleagues. So in thinking about what to talk about today, I thought I'd address the challenges for clinical researchers. So firstly, the poor cousin story. Who should be doing clinical research? Why should you do it? How should you do it? And how to achieve that very hard to reach balance that we've heard about. So yes, I do think that clinical research can be regarded a bit as the poor cousin of basic science. The degree of a PhD is really your entry level to becoming a basic scientist, and your clinical researcher comes along with their Bachelor of Clinical Science. And this really jumped out to me when the L'Oréal UNESCO team sent out a French film director to film me for my little 3 minutes of fame. And he said to me, when when did you want to become a scientist? And I said, well, until about 5 years ago, I didn't realise I was one. And I don't think that clinicians do think of themselves that way. And I put it to you that it's something that we should be doing. So why is clinical research undervalued? It's often perceived as less scientific. You certainly can't get a publication in Nature or Science that is pure clinical research. The outcomes are often far from black and white. Well, I think this is partly the clinical researchers' fault, and maybe at times we're less rigorous in our approach. We use less scientific methodology. But it's partly the nature of what we do, and it's very hard with the very complex organism called the human being that we work on to get to all the levels of the human being. And we heard before about the psychosocial impact of the disorders that we're dealing with and how you can work at looking at that at a basic science level, but it takes very careful planning and collaboration. So it's partly the nature of what we do, and often I think our conclusions may be poorly defined, so we need to think about this in a more rigorous manner. So some people would say, why bother? I think that clinical research makes an extremely important different way to the way you look at your world as a clinician and as a researcher. I think it makes what you're doing every day much more exciting if you can put it into a research context. And it also, very importantly for me, makes a difference to not just the patients that I get to see every day, but patients that I can touch and their families around the world, because your research can have global impact. It can certainly change practice, and by that, and to that end, improve outcomes for patients and families. So when you think about clinical research, you think about physicians and surgeons, so medical graduates, but I really wanted to highlight that clinical research is the province of all clinicians, whether you're a speech pathologist, a nurse, a physio, a dietitian, a psychologist. I could go on for hours, but there are many, many clinicians out there that should be thinking about clinical research in what they do every day. So then, is clinical research part of your day job? Is it something that you should do? Well, you can only do clinical research if you have those skills. So you have to be a clinician, and that takes a long time. And when I reflect upon it, it took 21 years of my tertiary education, including 2 sons, and a lot of clinical work, which is a long time. But what I'd like to know now that I wish I knew then was enjoy the journey. Don't worry so much about the end point. You'll get there, and there's lots to enjoy and try and balance on the way. One of the things I think that's very important for the clinical researcher is that every patient you see is asking new research questions, and you have to see how they can impact on your scientific understanding. The other key issue is that you've got to ask the right questions. So some questions might be vaguely interesting, but the answer may not be that important. So you've got to focus on questions that are worth answering. So is it your day job or is it a new job? And I think that in most clinical courses these days, people get a little taste of research. But that's it, and that's just a taste. It's not really research training. And I think if you're going to be a clinical researcher, you have to do high-level training, which typically involves a PhD, and this is a new career. I think that I and many physicians and other types of specialties in medicine, you come along and you think, oh, I should be able to do this. But there's no reason why you should be able to do this. Yes, you've studied for 12 years, but you haven't studied how to be a researcher. So you can't expect you know how to do it. You have to find a really good mentor or supervisor to help teach you. And to guide you along the way. And so one of the things I always tell my students as they pale is that every paper needs 20 drafts, and that often mine take even more than that. So clinical research, as you've heard, takes time and dedication, and it's not just an hour a week. It needs a lot more input than that. I think for clinicians it's important to develop the scientific thinking, and that's part of the PhD, but for those that go into clinical research and haven't done a PhD, you need to develop the typical scientific framework, and people often don't give this the full credit, with hypotheses, aims, methods, results, conclusion, and most importantly, the skill of critical analysis. So what can you do as a clinical researcher? Well, I think it's really important to think how you can take your work to the next level. And by that I mean the clinical observation, but then go on to think about causation, mechanisms, and then we have a very important role in translating that back to the patient. Now, for example, just to show how that worked with the autosomal dominant nocturnal frontal lobe epilepsy, that was the clinical research. The molecular geneticists found the mutation in the nicotinic receptor. The mechanisms have been studied by physiologists now for many years, and the more they know, the more complex it seems to be. But the clinical research and the molecular research have already informed diagnosis and treatment. So patients are diagnosed today and put on the right treatment because of this research. But I hope what I've shown you so far points out that a very important issue in 2012 is team science. You can't do research on your own. You have to work in teams and you have to find good collaborators. And as Elizabeth said, they all bring different views to the table, and those views inform yours, give you new ideas, new ways of looking at things. And those, so we could start with clinical research, go to our basic science colleagues who might do in vitro studies, but then you have to find other colleagues that are very happy to do in vivo studies because they can certainly inform our human science further. And then other colleagues still can, such as imaging scientists, can give you a lot more information to basically come back to help the patient. So we've heard a little bit about this already. I think it is important that women and men think differently. I think it's a wonderful thing, and that we can bring different things to the discussion as women, different ways of looking at problems, different problems to address, and therefore solving different questions. The other thing is that lots of people believe that there should be a eureka moment, that you have that magical discovery that's going to change science for the world. I don't think that happens for everyone, sadly, but I do think you can have lots of little eureka moments that can make a difference, and that we shouldn't poo-poo those either, because they can certainly take science forward. Yes, I saw this recently and I thought, and if you listen to Elizabeth's example and also our one with the focal epilepsy, often when you suggest something, first it's ridiculed, secondly violently opposed, and thirdly everyone says, well, that's obvious. That certainly happened to me with some of our research findings. I thought I should emphasise the importance of scientific output, and that means publications. Research is funded by the community, often the NH and MRC, but there are many other funding bodies. And we have an obligation to publish it, so not just to do the research, but to get it written and submitted and in press. And this even pertains to negative output. Doesn't necessarily mean it has to be at the same journal level, but it should be available so that the experiment is not repeated and resources wasted. Now, this is probably very self-evident to the basic scientists here, because careers are dependent on ongoing funding, and that depends on research output. But clinical researchers, I think, sometimes can just say, oh, it didn't work out, and not take it to that final point of publication. So in the last few moments, I thought I'd touch on that wonderful issue of trying to achieve balance. And I think that is really hard. And I think that is harder for women than for men in science. And it's because we're trying to balance a high professional level of work and output, and then go home and we tend to manage the family. Fortunately, in 2012, men are more enlightened. I think this is much more, and women, I guess, are both more enlightened, and we share responsibilities more. But you still see most of the responsibility fall to the woman, and I think achieving balance is a challenge. Everyone in the audience who knows me will know that I'm not an expert on achieving balance, but I'm sure that some of the pointers that we've heard about part-time work are very encouraging. I think you have to work hard to succeed no matter what, and science needs passion, and I know that all the people speaking today will have that, because they wouldn't be here if they didn't, but it's one of the most exciting things you can do in terms of a career. I would encourage you to have balance and to try and have other things in your life that can give you perspective. And I think having a family is one of those things. It doesn't have to be, but it certainly keeps you grounded. And I'm actually all in favour of multitasking, but my husband doesn't believe in it. So in conclusion, I'd like to, I hope I've shown you that clinical research offers exciting possibilities and opportunities that it's up to us as clinicians to recognise those patients that raise scientific questions, to design experiments to answer those questions, and we often find surprising insights that lead to important biological findings. We need to translate our clinical research to the bench with our basic science colleagues, lots of discussion and planning and working together. And then we have a very important role in trying to translate that back to the bedside to implement change for our patients. And I just wanted to leave you with that image saying really that I think anyone can do what they like. You just have to be very focused and keep your eye on the ball. Thank you very much.
Thank you, Ingrid. So now we move along to our final speaker for this session, and I'm delighted to introduce Professor Judith Whitworth. And Judith is known to many of you, I'm sure. She's also a graduate of the University of Melbourne, and Judith is the Director of the John Curtin School of Medical Research and Howard Florey Professor of Medical Research at the ANU in Canberra, where she heads the blood pressure unit, or high blood pressure research unit, I should say. Judith is currently an ambassador for Canberra and an ambassador for women, and she was Telstra ACT Businesswoman of the Year in 2004 and 2004 ACT Australian of the Year. So Judith is also a Fellow of the Royal Australian College of Physicians and has practiced medicine and researched extensively both here and overseas. And I'm sure many of you will know that she was chair of the Medical Research Committee of the NH and MRC for a number of years and past president of the Australian Society for Medical Research and the High Blood Pressure Research Council of Australia. And she's also chaired a number of international committees such as the World Health Organization Global Advisory Committee on Health Research and is a member of the Global Forum for Health Research. She has many prestigious appointments and has previously been Australia's Commonwealth Chief Medical Officer and Professor of Medicine at St George Hospital at the University of New South Wales. Judith has been made a Companion in the Order of Australia in 2001 for her service to the advancement of academic medicine and as a major contributor to research policy and medical research administration, both here and internationally. So to finish our panel of speakers, I invite Judith to the podium. Thank you.
Thanks. My task today is to talk about mentoring and leadership. So there are no PowerPoints, no dot points, and there are very few facts. I did go to the literature, but I found it was dominated by jargon and theory and a little light on evidence, and I'm sure you can all look it up for yourselves. What I do want to do is to give you a few personal views and anecdotes, so those of you who wish can close their eyes and fantasise or nod off as you like. Now, first of all, mentoring. Mentoring means to advise or train, and a mentor is a wise and trusted counsellor or teacher or an influential senior sponsor or supporter. The mentor's the teacher and the recipient is a protege, or horrible neologism, a mentee. My remarks are directed to those of you, and I hope all of you, who will mentor. But for those who wish to be mentored, no relationship you choose, apart from your dog and your life partner, is going to be more important. So choose wisely, and it really is worthwhile putting a lot of effort into choosing those people who are going to mentor your careers. Once you've committed, I think it's important to remember that in professional as in personal life, you tend to get out of relationships in proportion to what you put into them. We'll come to leadership shortly, but for leaders, success of their team or the individuals in it is their success also, and mentoring and leadership are intimately involved. People, of course, tend to learn far more from example than exhortation, from which it follows that you'll learn far more about mentoring and leadership by doing it rather than listening to a talk from me or anyone else. But to continue, I think it's very important for mentors to promote not just science, but scientific values, and those values of good conduct, scientific honesty, and unimpeachable ethics. Mentorship is far more than just training and encouragement. It's active promotion, and generous active promotion. For example, authorship. Could a postdoc warrant senior authorship in your place? Could one of your team take that speaking invitation? Would your team members benefit by serving on institutional or other professional committees, which are good networking opportunities? Would they benefit from learning about community engagement, from talking to schools or service clubs or talking to the media? Are there administrative tasks they can do, boring but necessary? And it's important, I think, for mentors to encourage training and professional development, whether it's by sending people on courses on how to communicate in science or leadership or whatever. Delegation's an art. It's a very important one for people like me who are very fundamentally lazy, and it's hard for people to develop skills unless they've got a chance to practice them. So that you know about my track record of teaching by example, I should admit that my daughter produces movies and music videos in Hollywood. It brings me to leadership. This is the action of leading a group of people or an organisation. Some people like to lead because they like control, and my remarks are not addressed to them. I prefer to discuss leadership in the context of achieving a goal. There aren't any simple rules because leaders and successful leaders differ enormously in style. And of course, leaders aren't necessarily forces for good. Stalin was on many levels a very successful leader until the day he died, but of course he was a monumental tyrant. So what we're interested in isn't so much leaders as good leaders. The qualities of a good leader are said to include being a role model, being willing to serve others, inspiration and dedication, a sense of mission, focus on results, working with others, a sense of optimism, the capacity to delegate and grow people, the ability to pick winners, self-awareness, and good judgment. But remember Elizabeth Bennet, in Pride and Prejudice, when Darcy outlined his many criteria for an accomplished woman, she responded, "I'm no longer surprised at your knowing only 6 accomplished women. I rather wonder now at you knowing any." I'd like to consider for a minute 3 of the most famous leaders of the early 20th century. They were Scott, Shackleton, and Amundsen. Shackleton reached 88 degrees south, but he turned back within reach of the pole so as not to endanger his men. Amundsen made the Northwest Passage in the Arctic and spent years living with the Inuit, learning about survival in polar conditions. And Scott and his men died after reaching the pole after a terrible struggle, only to find that they'd been beaten by Amundsen, by bad weather, bad luck, and it has to be said, by poor planning. A few years later, Shackleton returned south in the Endurance for it to be crushed in the ice, and in one of the greatest stories of survival ever, he eventually saved every one of his men. So to look at those 3, Scott was an English naval officer and gentleman who put the goal first and ultimately sacrificed himself and his men. Shackleton was an Anglo-Irish Chancellor who put his men first and sacrificed the goal. And Amundsen was a Norwegian loner who succeeded where they had failed through meticulous planning and execution. All great leaders, but in very different ways. What about women as leaders? Well, if you think about the ability to inspire others and a sense of mission, it's hard to go past Joan of Arc. She was an illiterate teenage peasant, but she led the French army to a series of remarkable successes against the English army in the 100-Year War. If you think of a sense of mission and single-mindedness, it's hard to go past Margaret Thatcher, and if you haven't seen the the movie The Iron Lady. I recommend it. My favourite story about Margaret Thatcher is when she was having dinner with the cabinet at the height of the mad cow scare. And the waiter said to her, "What'll you have?" And she said that she would have British beef. And the waiter said, "What about the vegetables?" And she said, "They'll have what I have." If you think about focus on results, I'm reminded of another movie, some of you may have seen, The Devil Wears Prada, where the Miranda Priestley character, played by Meryl Streep, is based on a famous real-life magazine editor, whose name I won't mention, who's a ruthless and completely unreasonable bully, but enormously effective in achieving the goal. I think it's important to beware of bullies. Quite a few leaders are bullies, and they can be enormously destructive of those around them and ultimately of their institutions. One characteristic I particularly admire is grace under pressure, and I think Julia Gillard's a very good example of that in the face of a very hostile and often dishonest media. And for other examples of leaders, I think you just have to look to the three women who spoke before me. Some 500 years ago, Machiavelli, in his classic The Prince, pointed out that in trying to effect change, you get lukewarm support, at best, from those who benefit, and vigorous opposition from those who won't. You can be a leader or you can be popular, but it's pretty hard to be both. I want to give you a couple of examples of some experiences from my own work. Prior to the introduction of the GST, the then Treasurer, Peter Costello, set up a three-person group, the Tax Consultative Committee, to decide how GST should apply to health, education, religion, and used cars. So we had less than 3 weeks to come up with a coherent plan. It was pretty intensive. I'll leave you to guess if I were the expert on religion and used cars. Anyway, I was of the view that tampons were not medical devices to be used in the case of illness and thus GST exempt, but sanitary products attracting GST, and that was what we determined. The money involved was about $3 or $4 a year, and that would be rather less than the compliance costs of actually making a GST exemption. Well, some of you might remember it hit the national headlines, and people doing feminist studies still send me angry letters. About the same time as Commonwealth CMO, I was actively trying to push an agenda for the use of evidence to inform policy. And a group of very committed people in the Commonwealth Department of Health had the excellent idea of taking health to the department, and they devised a plan for a Department of Health expo: displays, stalls, interactive materials, expert advice. And they showed real initiative and enthusiasm. The first I heard of it was when an advertisement for the expo appeared on my desk along with every other desk in Canberra. Much of it looked great, but it also featured a range of alternative therapies for which there is and was and is no evidence whatsoever. So there was a dilemma: pull the plug on part of the expo after so much well-intentioned hard work and reinforce all the stereotypes of medical authoritarianism and patch protection, or run with the fringe material and resile from the requirement for evidence, allowing alternative practitioners to ask why, when the department was promoting them, the government wasn't funding them. I chose the latter and again copped a fair bit of flack in the press, but we held the line and eventually it settled down. And I think the good news is that that push for use of evidence to inform policy is now taken off, not just in Australia, but worldwide. I think we're about 15 years into the 30-year cycle. The statement that you can do anything as long as you don't care who gets the credit is variously attributed to Edith Cavell and Harry Truman. In my experience, it's spot on. You need to be very clear whether you want the outcome or you want the glory. To paraphrase Lao Tzu, but if a good leader, when the work is done, her aim fulfilled, the people will say, "We did this ourselves." For the younger women among you, what does demonstrating leadership mean when you're not in a senior leadership role? Not yet. Leadership in this context may be actual or it may be potential. It may be leading by example through job performance, through innovation, through taking on responsibility, through making things happen. Some of you may have seen the famous old movie, The Wizard of Oz. As a kid, I really loved it. The heroine is a girl called Dorothy, and she and her little dog Toto are plucked from deepest Kansas by a tornado, and they find themselves in the wonderful land of Oz. And they make new friends, the Cowardly Lion, the Tin Man, and the Straw Man. But despite the wonders of Oz, Dorothy and Toto want to go home. The lion wants some courage, the Tin Man wants a heart, and the Straw Man wants a brain. So they follow the yellow brick road to find the wonderful Wizard of Oz who can make all their wishes come true. They have lots of adventures, and finally they find the wizard, a disembodied loud voice behind a curtain. But before they can have their wishes, the curtain falls away and the wizard's revealed as a middle-aged man with a megaphone, a fraud with no magic powers at all. And Dorothy and Toto and Lion and Tin Man and Straw Man realise that if they're going to get home to Kansas, or be brave, or get a heart, or get a brain, then it's up to them. And they go off and do it. So my message to all of you about mentoring and leadership is simple: go and do it. Thank you.
Thank you, Judith. I'd like to invite all our speakers up to the chairs up the front so that you can see the audience. And I'm sure you've had lots of wonderful thoughts in the audience, and now's your chance to ask some questions, to make some comments. So if our speakers can come down here to these chairs. Who would like to begin? And we've got microphones here so that you'll be able to ask your question. So as the speakers are getting organised and you're thinking of your questions, I'm sure you were all struck by various things throughout the talks, but one of the things that I was thinking about, one of my most favourite books is the book by Georgina Ferris, the Dorothy Hodgkin, A Life. I don't know if any of our panel members have read it, but Dorothy Hodgkin was, I think she's the only female British Nobel laureate, and she won the Nobel Prize for crystallography. And in her final, I really do recommend it to anyone for inspiration. It's a fabulous book. And in one of her final talks, she's Chancellor of a university in the UK, I think it might have been Bristol or something like that, and she was addressing the final group of women to go forth from their studies. And she said to them, "I at least hope some of you live simply and do serious things." And I've never forgotten that, and I kept thinking of it when you were talking about Dorothy's words of wisdom. So who would like to ask a question? Don't be shy. Yes, we've got some hands up, and there's another one up the back if we want to get another microphone. How many microphones have we got on the... Just one, okay.
A question about mentorship in general. I'm a PhD student at the Paediatrics, and we've got kind of an institutional mentor program, which is when we select our mentor, it's really based on support on an academic sense and not really broader issues about career and things like that. And I'm just interested to see what you'd think of, like, how do you approach finding a mentor who's got the kind of characteristics that you would like to follow through with your career, maybe like external to your institute or the department that you're studying in? How do you kind of get that ball rolling and approach people about being your mentor if you're not directly associated with them on a day-to-day basis?
Okay, about mentorship. Is that directed to anyone in particular? No. Okay, who wants to have a go?
Well, I've got the microphone, but I'll pass it on pretty rapidly. In my experience, most people, when approached, are pretty happy to help. And it's like all these things, you might get some knockbacks, but someone will say yes. And so don't be afraid to identify someone you believe would like to act as a mentor and approach them.
Thank you.
And I think not just one mentor. Sometimes there are certain people who are terrific at some aspects of your career, but may not necessarily have a terribly useful perspective on other aspects. And feel free to ignore what they say too.
I'd like to echo that and say, look for mentors in other fields and other domains. people that you admire, but also people that are nice. So I think it's worth asking their students or the people that work with them what they think of them, because that will give you a bit of an idea, and then set it up. And if you want to have just a general chat, say a cup of coffee or something, that changes the setting, and that helps.
Yes, I guess it's all been said, but I think approach people you see sympathetic and ask them or tell them your problems, and if they are truly mentors, they'll respond. If they're not, then go and find someone else.
Good advice. So next question I think is right up the back. Okay.
Hi, I'm Katrina Williams. I'm Professor of Developmental Medicine at the Royal Children's Hospital Campus, University of Melbourne. I want to ask a question based on a comment from my son. He's not here, so I can't embarrass him. But at the time when you were awarded your Nobel Prize, you appeared on the ABC News, but I think you were close to or just after the sport. And my son was outraged at this and where Australia was placing science in the community. So it's not entirely a women in leadership or women in science issue, but where do you think we are in the 30-year cycle for change of the place of science in Australian society?
Okay. Well, I'm sorry I interrupted the sport.
That's not why he was outraged.
I would love to see... So we look at sports in Australia and say, you know, this is something that's done really well. It's a national passion. Why don't we think of ways that we can capture some of this for science? I mean, there is an aspect of science, a little bit sporting. I mean, being a little bit frivolous here, but also trying to think about maybe there are things that we can learn from sports in sciences and try to draw on that sort of excitement. And I don't mean, you know, science competitions or something like that. I don't know. It's an interesting thought, because you know, sports in Australia is so highly developed, as it is in many countries, but particularly achievement in that is very valued. One explanation I was told about the value people place on sport is that it's perceived that anybody can do it, and you know, achievement in science and so on is sort of seen as rather exclusionary, that not everybody could do it. So I think that involves really making sure that access to good education in science, so that, you know, all the schools have terrific science education, makes it seem not so sort of exclusionary. Because, you know, sports is about achievement and science is about achievement too, and so it's they have a lot of similarities. So I think we should try to be imaginative about that.
Thanks, Judith.
I've got a little confession to make. A few years ago, I was on an aircraft, and I was sitting next to a member of Parliament. I won't name him, but he looked at me and he said, "The only woman I've ever seen who starts reading the newspaper from the back page." So I actually love sports of all kinds, and I don't think it's just a problem for science in this country. I think it's a problem for most sorts of intellectual activity, and So I don't think science is unique in that regard, but we need to do a lot more. We need to get out there and be selling not just science, but I think rationality, the use of research and evidence in policy in everyday life, and we need to be out there combating, I think, the terrible contemporary tendency to cry logic and rationality, and to make megaphones available to morons.
Okay.
Present company excluded.
Yes.
But I think maybe, can I draw a little on that and say, we tend to sort of think, well, I suppose we should try and make it more interesting and alluring, that, you know, because you know, rather than saying, well, you're bad people, you're not doing science right. I mean, that's true, we all know it's right. But I'm also thinking, I think we should try and think about ways that we make science interesting. I've just been talking to secondary school children in various capitals, and I really thought it was important to try and make it obvious that science is interesting, doing science is interesting, and not only just being a scientist, but actually, as you say, having all of those ways of thinking critically gives you a lot more you know, ability to do things in ways that go far beyond science itself. So it's sort of up to us in order to make it, to lure people to it, rather than tell them they shouldn't be doing something or other. So I think we have to take a very active role in trying to make people think this is a good thing and as fun and interesting as sports.
Just wanted to add, it's cultural change that we need, and you've outlined that beautifully.
My only problem with science, talking about, is that it's such a broad subject. And really this was the problem I had beginning, because was I going to do science? What is science? Mm-hmm. And I broke it down eventually into lots of different subjects and chose one I liked. But I think when you're talking to children about science, again, perhaps it goes over their heads a lot of the time, or they don't really understand what you mean by science.
Mm. Okay, thank you. Next question. I saw a hand up. Someone has the microphone? Oh, just behind you. Yeah.
Thank you. I'm Sheil Riley. I'm Professor of Speech Pathology at the University of Melbourne and the Murdoch Children's Research Institute. Ingrid, thank you for emphasising how broad medical research is, and that it's actually more than medicine. I appreciate that comment, and I think that's really important for the future health and well-being of the country, that we think about it being broader than that. I wanted to make a comment about women and child caring. Having got to this stage in my career, I thought I'd put all the caring behind me, but I think there's another challenge for women and for men, and I'd like it to be a bigger challenge for men than it is, and that's caring for elderly relatives and taking on those caring duties later. And I think they do tend to hit women, and they hit women at very important parts of their career when they're often moving into very senior positions, and I'd really welcome your comments on that.
Thanks, Sheena. Who would like to take that challenge? Judith.
I'd just like to make a comment on the first part of your remarks about how we've moved on from medical research to understanding the importance of health research, and I think the next paradigm shift is going to be talking about research for health, because health is impacted so much by other areas of society, education, transport, all of those things. So I think that what you say is very important, that we've moved on from a narrow view about where the research should be to a rather more holistic view, which is more likely, I think, to impact long-term on well-being. As for carers, I wish I had some answers.
Research has touched on that in the sense that the health impact of being in those chronic situations, of being a primary family caregiver, has, you know, quantifiably read out in telomere maintenance. And that's been very interesting because, as you're saying, it started to reach out into questions of health policy now, and you know, and to think about that in itself having impacts on the health itself of the carers, you know, starts to reach into policy issues that I think we're just beginning as a society to really grapple with. But to have it taken seriously and not just sort of have the burden thrown on, oh, well, you know, the women of the family just always do this. And that's they're going to be, you know, I think you're touching on that this be recognised that it's a serious responsibility, that support for this really is important in various ways. And thinking about how families, you know, sort of problem solve and try and be able to deal with this is really just an emerging thing as we go into this big, you know, epidemiological burst of what's happening, right? As the demographics are changing, this is what's the big sort of tsunami waiting for us in so many ways. So I think we have to grapple with it.
Your comments are particularly pertinent to my last fortnight because my mother-in-law is 92 and has a fractured pelvis, and we took her home on Saturday, and she's showing some early cognitive decline. And she has 4 wonderful sons who are not remotely medical. And so I'm trying to juggle this and not run it and let them look after their own mother, but help support them in that. And I think it's really hard. It's very hard with our careers to find time for that as well, which is extremely important, but you can't just stop work and stay home and look after her. And I don't think there's any framework in our society for this at this stage. So it's something we need to think about.
Thank you. Okay, we've got a question down the front. So the microphone has... Oh, it's gone. Oh, there's one up here. Okay, we'll take this question first and then if you want to come down the front. Okay.
Hi, Crystal Evans from the Walter Eliza Hall Institute. There's been some great suggestions today about discussions on ideas about how to bring about institutional change. So the examples of access to childcare, the example of full-time versus part-time work. How do you think we, as young researchers, and not just women, men in the room as well, how do you think we can help bring about institutional change from our level?
Great question. Easy to answer. Ingrid, no. Ruth.
I don't know that part-time work is such a novel project to try and organise. I worked part-time through a large portion of my career, 10 years or so, and what you actually do is you're paid part-time, you attend where you work part-time, you do what you have to at a desk part-time, and then you go home. You might have Friday off or holidays off, as I did, but your part-time is boosted by voluntary work you do at home, which is a lot of reading, a lot of writing. So you keep your career going in that way. Now, this is what I did. I'm not necessarily defending it, but it worked for me. And I think that there is scope in NH and MRC or granting bodies to fund part-time workers. If not, then we're fortunate at the Royal Children's to have funds we can apply to. And I've always found a great sympathy for that. So.
See if anyone's got anything to add. Ingrid, yes?
I think a way to look at institutional change is for women to get to the top of the institutes and to remember the hard time they had on the way there. And sometimes I think in the past the women have got to the top have sort of forgotten the hard road. And I think that it's incumbent upon us that as we get to become more senior, we help to influence policy so that there are ways for people to succeed. And I was really interested to hear what you said about the part-time scientist who had 4 children and managed to do incredible science. And I'd love to hear about the journey for her in terms of funding, job security, those issues, because whilst we all say NHMRC should take account of the women and their time off having children, it's not just the 6 months or 12 months that you have the child, it's then the years of wonderful motherhood that needs extra time. And how can you put that into the NHMRC little equation?
Well, I think institutional change requires a lot of hard work, but I think if there's a cause that you want to push, then you push it at all levels within the institution, you try and bring others with you, you bang on about it, and so on. And as in most things, I think the way to achieve change is by getting out there and communicating what's needed to others and not letting up.
Thank you. I mean, it does sound to me like there's no doubt that a lot of time is required no matter how you do it, whether it's flexible, whether it's not flexible. But would any of you have any comments on, is there an easy way, or does it just does take time? Is there a shortcut?
What's wrong with
what is
spending time on it? I mean, we like it, right? I really think that we do worry a little bit too much, like, well, will I be able to go to the films and restaurants and things like that? I think it's okay to have waves of great... I know I did nothing but family and science for a number of years, and we'd done all our travelling and stuff for quite a while before and seen lots of films, and now I see lots of films and go travelling. But the thing is that if you really feel this is important work, time is not, of course it's going to be hard work. And it is true. It is true. It is hard work. But both of them feel very worthwhile. Yeah. And so I think this idea that somehow everything should go at some very easy little pace, I don't think that's actually the way you're going to get things achieved. You are going to have to throw a lot of yourself into it, but that's why you're in medicine or in biomedical research or something. It's because you really do feel more than this is just sort of, just a way to bring home a paycheck. I think most people here really are very passionate, as this title of this symposium is about, says. And I think we shouldn't... I think this part-time thing, it's always seemed a little bit as though, well, okay, that's just, women will, and sort of it implies a second best. What I think we have to do is try and make sure that the part-time idea is not seen as second best, which is why I very much liked what the head of this astrophysics group in Sydney actually said, which is, he said, this is just a priori to advertise jobs as part of full-time, and he said, and for men or women too. So it isn't seen as somehow, the women are always the one taking the second best, the part-time. That's what I'd like to really see. Can that be made just sort of more the norm? Because we have seen that part-time does not preclude having terrific work done. And that does mean things like the NH and MRC recognising that the timing and the trajectory of work and productivity doesn't have to be all on one time frame too. So again, not recognising when people are working part-time, the way they'll produce publications will be different from when they're on a full-time trajectory. But that doesn't mean they're worse quality, quality, not quantity.
Thank you. We've got another question just... Oh, that's okay. And then behind you, do you mind, Leanne, if behind you and then you?
Dr. Rachel Dunlop. I'm a postdoc currently in between jobs. Professor Whitworth, I was very interested to hear what you had to say, particularly with respect to decrying logic, which is a common attitude now. And I'm especially interested in how this, as a scientist, I'm very interested in vaccination science, so I'm particularly involved in encountering this. And there's also been a rise in the role of postmodernism in science and medicine lately, where people want to be their own, they want to control their own destinies. So with the rise of the internet, everyone's an expert. I just wonder what you think is our responsibility in addressing this rise in science denialism. Should we do something about it, and what can we do about it?
Thank you.
And the answer is yes, we should. In fact, I'm part of a working group of the Academy of Science at the moment, which is trying to produce a document on immunisation, analogous to the excellent document that was produced on the science of climate change, to communicate that to the public. When I say the public, the level that's chosen is the Year 12 level, so it's for an educated public. But yes, I think we've got an enormous responsibility to be out there speaking out for rationality, for evidence, for research. And I think in these days with social media so prominent, much less censorship, the moves by the rich and powerful to take over the media, the abuses of power by the media that we've seen in the UK and internationally, are a real worry. And I think all educated people have a responsibility to be out there standing up for rationality and for truth. And indeed, my memory is that it was Cardinal Newman when he talked about the aim of a university education who, to paraphrase, I think, came down to the view that it was firstly to create good members of society. So I think, yes, it's a big responsibility for us.
Okay, Leanne.
Thank you. Leanne Tilley, Professor of Biochemistry here at Melbourne University. I was struck, both Elizabeth and Ingrid touched on this, the concept that women have different ways at looking at a research problem from men. And it was also interesting that difference between what you might call the hard or basic science type research and the clinical or humanist type research. And my feeling was from what you were saying is that you thought the women were really, really good at the humanist stuff and clearly were also rather good at the basic stuff. I think Elizabeth started basic and perhaps has moved more humanist, and Ingrid started clinical and perhaps moved more basic. But I was just wondering, that's a really interesting divide, the way to think about things, and whether you think that women really are better at that more humanist, some research that's being driven by helping humanity as opposed to achieving a scientific goal. I just thought it might be nice to explore that a bit more.
Okay, thanks.
Yeah, I want to very much avoid what I think is sounding like a stereotype. I don't think it's a divide. I think it's interesting that sort of historically that was where people, in my experience, people who came and approached me as collaborators happened that they were women in this area. Now there are men in the chronic stress area, very, very accomplished, researchers in that area too. So I don't know whether it was partly the sociology of science that women approached me as a female scientist in the basic science. So I would hate it to be a divide. I really would. I think subject matters for science sort of happen when people feel the problem becomes approachable by scientific methods. I think the enormous complexities of many of the humanist aspects, the social and so forth, have deterred some scientists. And this is one way one could ask questions in ways that could speak sort of to both worlds. But I don't want there to be a divide. I think that we want to, humanism belongs to men and women too, right? I think it was just sort of interesting that this was how it happened in my case, but I think I didn't want to posit more than the idea that things get asked in ways that might be different, because of men's and women's experiences, but I don't think that should be forever or the way it is. So I'm not being very clear about my point, but I think the 2 points are both men and women have done very humanist and related research. It's just historical or contingency that they asked questions they did, asking me to be a part of it. It's a very interesting thing, and I ponder it, but I don't have good answers. But I think the right, the wrong, the hard science, the not so hard science part, that part of it, I don't think is actually really gender related, although there may be stereotypes about it. It's interesting. I may be wrong. Looks if you want to comment.
Yeah, I just wanted to make a comment about hard versus soft. My thinking about this was really radically changed some years ago when a qualitative researcher put to me that you didn't need to be smarter to do hard science. In fact, one could argue that the fewer variables there are, the less smart you need to be, the easier it is. And that in these, the more complex the area, so as you move from molecular biology through to clinical studies to epidemiology to complex studies of societal functions, then it gets really, really challenging. So I think that's a useful way of thinking about hard versus soft. Of course, when you're thinking men versus women, I have to say how good women are at multitasking.
Thank you. I'm pleased you said that. Yes, I also didn't mean, I think that there are very good women basic scientists, very good women clinical scientists and good men. What I really meant was that I think women look at things differently from a different aspect, and I just think that they can therefore ask different questions and maybe get different answers or go down a different research path. And I think that's very complementary to men. And the absence of having enough women in high levels of science has been very apparent, has been discussed. There certainly were no female mentors in my world. And I think that we shouldn't underestimate the fact that both sexes bring different things to science, and they're both equally valuable.
Okay. All right, over here.
Thanks. I'd just like to follow on from that. My name's Misty Jenkins and I'm a postdoc at the Paediatrics. Also proud part-time scientist and mother of a baby, and trying to juggle it all at once. And I guess I'm becoming increasingly concerned about the short-term political funding goals that perhaps the government has, and putting pressure on the NH and MRC to actually focus, to fund projects that do have a clinical focus, and perhaps are not giving enough weight to what we would call the basic science or the discovery-based kinds of projects. Do you agree that this is a cause for concern, or do you think that just with limited resources, you know, it's just the way the cookie crumbles?
Thank you. Who'd like to take that one?
I'll be very aggressive and say, what better way to fritter away your money by having tiny little short-term things that nobody can actually get their teeth into, and you know, and so therefore nobody actually does anything more than slightly incremental steps because they're limited by the short-term goals. Woo! No, I'm just sort of trying to be a little over-provocative and saying, no, I think research should be invested in ways where people can get their teeth into a problem. If that means funding for 3 or 5 years or something, 5 years probably is better. Where I'm doing my research over the decades that have just passed in the US, I mean, that's where I've been doing it. The politicians certainly have extremely short time frames, but 5-year grants have been the norm for decades. So I don't think that that is necessarily tied in. You know, NIH budgets, you know, they've been 5-year grants for a very long time, 4 and 5 years. And we know the politicians have, you know, a 2-year cycle in Congress and so on. So I think we really shouldn't think about funding research in ways that limits the kinds of research projects that people will do. There's certainly a place for very clear goal-directed, you know, specific projects, right? But I think that actually doesn't make use of all the talent that is in the biomedical and medical and clinical research communities. That's my 2 cents worth. Would you like to speak to this?
No one else?
Nobody.
You might have to get people at afternoon tea. Okay, we've got another question up the back in the blue. Do you want to ask something? So we'll have time for probably one more after this, and then we'll have afternoon tea.
I'm Grace Chu. I'm a breast surgeon and a cell biology, cancer cell biology researcher at St. Vincent's. Firstly, I want to thank you for putting on this wonderful afternoon. Certainly, there's a really high caliber quality of women researchers here, and it's really inspiring to hear from all of you. I guess my question was kind of related to how can we encourage women researchers of high quality to continue going on with their work and to continue working in Melbourne and Australia rather than losing them to, I guess, other centres in other parts of the world where perhaps, you know, there's a bit more financial or structural infrastructure that's, you know, available and more amenable for women in this sort of working environment. Okay. How do we encourage women to continue doing the work?
Can I speak to that?
Yes.
But I think you just said it. A kind of career structure in which you have a year-to-year contract and uncertainty. I mean, as you said, we all have to live with uncertainty, but we live with that in ways. When we do research, we're putting ourselves into the realms of uncertainty all the time. But as a career structure, to have, you know, year-by-year contracts, and again, this speaks to this issue of having longer grant cycles where people can get their teeth into problems, and then having infrastructures that don't just, you know, treat it as sort of temporary year-to-year contract labor with no certainty. I think that really isn't conducive to people wanting to have careers like that. So I happened to be in the US where I had my career, and I felt I could have a career structure, that there was an infrastructure and that there was a granting situation that had at least something that didn't make you feel you were living from hand to mouth. And it wasn't that you were sitting back thinking this was a sinecure, far from it. I mean, you're absolutely out there competing all the time, working very hard. But you sort of had a feeling that the structure was there to try to make you succeed, not to sort of try and undermine you, right? So I think there's a lot that, if you feel that that's not true here, then there's a lot to be done. Do you want to speak to that?
I agree with you. I think it's not true here. I don't think we have good infrastructure for careers for women in science, and it needs to, again, maybe go to more senior leadership by women and more insight into how to fix the problem, more policies.
Thank you. Ruth, do you want to add anything? No? One last question. We've got
men too, right?
Now, of course, everyone will put their hands
careers.
up.
We're talking about science career structures.
We have a question right down the front for the last one. And then I'll ask those of you who haven't had a chance to ask your question that there'll be time over afternoon tea to discuss. Okay.
Hi, my name is Maria. I'm an honor student in St. Vincent's Institute. Maybe my question is a bit overlapping with previous question. What other factors do you think that contribute to women living in science, especially in the medical research, and what the how to reverse that problems if you have a suggestion?
So why do women leave science, and how can we stop it? Why do women leave science? Why are they leaving scientific?
Other than family matters.
Other than family matters. Does anyone want to comment on that?
We're asking the wrong people.
I did have that thought myself. I thought, well, you sort of hung in there, or for the. So
well, I think,
why did you stay? Why did you stay?
But I think your question is a very good one, because I know the pattern in the United States, I think, is that the loss of women from, the 50-60% of people who are in medicine, in biomedical sciences, who are women, happens at the stage of the postdoctoral period. And so one has to say, well, what's happening then? Because they've performed equally well to men and in comparable numbers, and they didn't suddenly become stupid overnight, right? And lose aptitude. So we have to say, what are the factors that make people make a decision for a job? And I think some of it is this career inflexibility, this how can I possibly do it when the only sort of stereotyped way to do science or medicine in a full-time sort of one-size-fits-all model. So that's why I keep thinking, if we think creatively about how the trajectories can go without having to have that model, people will not feel so overwhelmed. But the young people look at us faculty, I know, and they say, you're insane. How could anybody in their right mind want to have a position like this? So the career structures look very daunting to young people. And as I say, I think a lot of it's because they're seen as somewhat inflexible, and they've seen one kind of successful model, but not enough examples of other ways of doing it.
Okay. So I think the final word was about flexibility, wasn't it? About diversity, doing things differently, and also a very clear message, I think, from the panel about if you're unhappy with how things are, don't just sit silently about it, actually make a noise. Rage, I think you said at the end, rage a little. You've got to choose when to rage a little. Don't rage all the time, that's not going to be good. But and pick yourself up afterwards. So I think we've had a wonderful beginning to the afternoon. We're going to have afternoon tea and then some more presentations. So I'd like you to join with me and thank our panel for a wonderful introduction.